So, good afternoon, good morning, good evening, everybody around the world, and welcome to the Phenomics webinars. For this Friday, we are welcoming once more Professor Menachem Moshelion from Israel. Hello, Menachem.
Hi, Philip. How are you?
Not too bad, thanks, Menachem. He will give his talk on functional phenotyping of plant responses to abiotic stress and shed light on standardization, screening, and analysis of your tomato population inside greenhouse and maybe also outside. Let me quickly, before we dive into a short interview in front of Menachem's talk, to your hosts today, which will be basically me from IPPN. My name is Philip von Gillhausen, hello. And my colleague from the University of Nebraska-Lincoln, our IPPN Academia Section Chair, Jennifer Clark. Hello,
hello
Jennifer.
Everyone. Yay, welcome.
So, coming back to topic, Menachem. We decided for today that we, instead of going the usual way and having several standard information being put up before your talk, we'll have a little interview to which you agreed to bring you a little bit closer to the audience. So for first, Menachem, I will introduce you just a little bit, and then we will dive deeper with our conversation onto that. So Menachem Moshelion is a professor at the Hebrew University in Jerusalem, or of Jerusalem. It doesn't necessarily need to be in Jerusalem, as I have learned on my last visit. And there he is working in the Faculty of Agriculture, Food and Environment on the Division of Plant Sciences, Genetics in Agriculture. This is correct, Menachem, right?
Right, absolutely right.
But I saw, and of course, we have known each other for quite a couple of years. You are also a co-founder of the phenotyping technology or phenotyping system technology provider Plantarray.
Yeah, this is
so. How come? Do you want to explain us a little bit how it came that you founded this company and what this company does?
Yeah, well, actually it was a necessity for me because during my research, it was more than 15 years ago, we had to phenotype, my primary work is with plant-water relations, and the cellular work is in particular working with aquaporins, water channels mutant and potassium channels mutant. And unfortunately, these crops had no visual phenotype, but you saw they have very bad yield, and it was very hard to work with those because we have literally hundreds of plants, but we could not get any numbers out of them. And we really look for a way to screen this water relation because we saw they sometimes look poor, maybe don't close stomata as good, but there was no high throughput physiological phenotyping back then. So we just started to build one. And you know, one thing came to the other. Then we started to work with an agrotech company named Evogene. And we noticed that after 3 months with our systems, we could detect mutant. Took them like 10 years to find. So pretty soon we understand that maybe this scientific system can also help others. So we raise some money. We start to understand how is this business work, and you know, thing get to other. And in the end, a private company came. So actually, the Hebrew University has the patent. And then it commercialized it to Plantarray that's actually built the product.
Yeah, and by now, I mean, there's a couple of years that went in since then. And by now, your system has acquired very positive reputation around the world. I hear from projects in the Netherlands, for example, where these systems get deployed as component of a top-notch state-of-the-art phenotyping centers. So you must be quite proud to have achieved something like this.
Actually, yes. Well, as I said, it was purely scientific based, and as you say, Washington now also will start working with it. I think the concept of working with non-image based system, but that gives you a lot of data from one end, but also help you to regulate your experiment. And this is the standardization I will talk about in my talk. I think that this thing makes it very unique. So you can really control the dynamics of the plant environment interactions and get the data at the same time. So yeah, I really hope it will catch more.
Well, and I just recently heard about a new center of functional phenotyping in Israel itself called ICOR. Can you explain us a little bit what this means, what this center is and where it is?
Yeah, so Israel established excellence core or core facility for phenotyping, which in this center we have 504 units of Plantarray that enable us to screen large set of population for many parameters like drought, salinity, nutrients. We can give up to 9 different cocktails of nutrients per each plant. So this center was established by the Israel government money, but it's open for the audience. So I put in the chat the address. So if somebody needs services, you can just click registered, and if you will find a way to bring his material or his seeds to Israel, he could get the service. We already had few people using this, I mean, out of Israel, people that use the service. You get your data via the cloud, via the internet, so it's very easy. You don't have to have big servers or something. So this is it. This is what the service does. It just gives you the opportunity to do screening for your either material or plants.
Sounds like a great opportunity, especially for people who do not have access to plant phenotyping systems and need any kind of data in this regards. So for everybody interested, be sure to check the chat for the link to the ICOR facility, and maybe you want to consider also entering the ICOR facility into our international phenotyping systems database on our IPPN homepage. This is being used internationally to foster collaboration and also, of course, enable the exchange of not only data. So be sure to check this opportunity. And let's move on to why everybody today is here, namely your talk. I'll quickly raise attention to some simple rules. If there are questions that pop up in the audience during the talk, please write them in the chat. Yeah, don't use this raise hand symbol, which is also another option which we are not using. If you want to make sure that your question is later asked in the discussion, then put it in the chat at any time. And Jennifer will keep an eye on the chat and collect all these questions for later on. Yeah, then I want to, of course, highlight that this webinar is also being broadcasted in China currently due to the daylight saving time shift. They are recording it and releasing it the next day, so people can look at it, view it at a reasonable time. And of course, once more, thanks for the technical support from the University of Nebraska-Lincoln. So, Menachem, I'm through with my introduction, so I will keep my mouth shut for now and leave the stage to you. Thanks again for being here. The stage is yours.
Thank you, Menachem.
Thank you very much, Jennifer and Philip, for inviting me. I'm very excited to present to you my talk. So I will just share my screen with you.
Ah, perfect.
I will make it a bit bigger. So you see my pointer?
Yes, yes, that looks great.
Okay, So with no further ado, I think Philip make a great introduction. So today I would like to talk to you about few topics. First of all, I would like to explain you the importance of phenotyping the whole plant-water relations. Especially if you go to crop productivity, the whole plant gives you something that it's usually hard to get by sampling a plant or just taking like one leaf or part of a root. Then I would like to talk to you about the crop productivity vulnerability trade-off, which is again a very big issue in predicting yield from crops. I would like to emphasize a bit about what is stress terminology, and I would like to suggest some options of using it because there is a big misleading in the literature. And I would like to talk about the phenotyping challenge of the plant-stress interactions. I will introduce the Plantarray platform, which we use, and I will also show some case study of how we could use this whole plant-water relation phenotyping in order to get yield prediction in a pretty accurate way. And when I say yield prediction, I mean in-field yield prediction. So plants doing photosynthesis for us, right? They do it for themselves, of course, but we use this photosynthesis for food, feed, fiber, and so on. In general, plants take water, and while transpire the water through the open stomata, they also absorb CO2. So we can say that as more stomata is open, the higher CO2 the plant's going to get, which is true. The problem is the ratio between the water and the CO2, which is in crops, it's huge number. It's 500 molecules of water per 1 CO2 on average. And if you look on this graph, you can clearly see it. On the x-axis, you see the water transpired by a plant. And on the y, you can see the grain yield. And you see clearly that it's a linear correlation. So if you can measure accurately how much water the plant transpired, you will get pretty good estimation of the grain yield. So if you can measure actually also the plant surrounding like light, in this graph we can see the radiation the plant gets, and also soil water content, atmospheric condition like VPD, temperature and humidity, altogether you can get a very nice estimation of the plant productivity, and you can even distinguish between more efficient like this C4 plant and less efficient. All in all, when we look on this graph, we can see that if the plant doesn't transpire too much, it will also don't have too much yield. And this, in fact, is a very important productivity vulnerability trade-off. And this is the subject I would like to talk about now. If we look on wild type, so we can say that evolutionary, this wild type develop some strategy to survive their environment. They produce, but they also need to survive this strategy, okay? So these plants are fitted to their own environment. Nevertheless, when we talk about breeding, breeding had very, or I would say a very important goal, which is improving the crop yield. This breeding process, and there are many papers showing it, mainly increase the plant size, increase the stomatal aperture, and also elongate the time of the stomata opening. So we can say that this plant or crop plants actually open stomata much more than the wild types. So as a response, they produce much more, but they are also more vulnerable to droughts. We can see this phenomena in many graphs. I choose one of Richards, a very famous Australian researcher. He showed the release of wheat cultivar to the market. You can see nice improvement of the grain yield on the x-axis and the year of release. But when Richards took the same plants and this time measured the stomatal conductance, he got even steeper slope, sorry. So the plants that transpire more also produce more. So if we look on this very simple chart, we can say that wild types or land racers are usually low yielders, but have a very good survival mechanism. So we will find them here, high survivability, but low productivity. Breeding actually pushed the plants in or toward the high productivity to this end of high yield, but it makes them much more susceptible to drought. The goal today of many breeders is to push the plants up here. So we have both high yield and good survivability. We call these plant idiotypes. Unfortunately, we don't have too much of those. But also, unfortunately, most of the crops we have today still respond to stress by both reducing yield and being sensitive to the drought. So today, this is one of the major agricultural problems. Modern crops are sensitive to drought, and that means that under drought, they will suffer a lot. So to summarize this part, we say that crop breeding actually opens stomata much more and for longer parts of the day, which in turn produce more. So crops which open stomata longer time of the day produce much more yield, but also have much worse water use efficiency because they lose water at this time. We can also say that these plants are risk takers because if drought starts, it will take these crops much more time to close stomata, and this is part of the explanation why modern crops suffer so much from drought. So many people, many scientists today try to improve this trade-off. I would say they try to push the plants to keep the high yield, but also to improve the water use efficiency toward this part. So you look in the literature and you find many definitions. We found drought resistant, drought tolerance, drought resilience. Now, what does this mean? So I would like to talk a bit about, or even suggest some terminology for this plant drought response. So usually if you look on a crop, usually it starts when the conditions are good, right? Normally growers start the field usually after the rain. But then if abiotic stress starts, let's say drought, the susceptible plant will just die. So we'll just call it susceptible. Some plants show escape mechanism, which means they will flower early just to like run away from the drought. The problem with the escape mechanism, we usually have very low yield. Many plants which suffer the stress, but at the same time, actually activating all kind of defense mechanism. So they can start recover from the stress either by activating this defense mechanism, or usually, or many times, you have also rain coming here. Either case, the plant will recover from the stress in a terminology I would like to call resilience. So the resilience is the recovery of the vegetative part of the plant. So the plant that suffers under stress will start to grow new leaf, new roots, new shoots. Now, if the plant has low resilience, it probably also will have low tolerance. So tolerance is basically the amount of yield that the plant produces in response to the drought. Some plant has nice resilience, but nevertheless, their flower or any fluorescent part was hurt. This is why they may have very nice resilience, so big shoots, but not too good fruits. Maize is a good example because if maize affected by drought, when it's flowering, we can lose up to 60–70% of the yield. Nevertheless, some plants has a very good resilience, so they have high resilience, which means nice recovery of the shoot and also nice recovery of the fruit. So we have high resilience and high tolerance. Resistant whatsoever is actually no stress at all. So we can say that the resistant plants will show similar yield to the non-stress control. In drought, we don't know any phenomena like this. There are some plants that are resistant to disease or resistant to salinity, but not to drought. Drought will always have some yield penalty. So these are the suggested terminology that I would like to use, and I will use them also during this talk. So if the breeder goal is to find the ideal stress response behavior, we must remember that we have to define the stress condition very accurately, because stress will be different in Australia, in Israel, in United States, and in Netherlands. It's not the same stress. So stress should be defined very clearly. And we must put a nice or the right control, which means we always want to compare the stress plant during any phenotyping system we use to a control with no stress, because the penalty is always the delta between the stress, the defined stress, and the control. And this is something which is very important to remember. So when we look on the funnel of breeding, especially in the pre-breeding, we are looking for a parental line with very good traits that could really improve our plants and help us find this ideal stress response. The problem with stress breeding is the time it takes. In this cartoon, you see 20 to 25 years, and this is not exaggerated at all, because looking for stress response, in particular drought, is very time consuming, and especially because you always have to do a feedback between the agronomy part and the crop physiology part, and really see that what you find is really stable and repeat himself for year after year. This bottleneck of phenotyping, this is maybe the major part of the pre-breeding process. So how to phenotype my plants and how to know that these are the right traits to go toward to the field. So the question many of us ask themselves is, when you need to phenotype, so where are you going to phenotype? I mean, in greenhouse, in the field, when to do it? When the plants are young, old, flowering, not flowering? What to phenotype? Maybe this is the million-dollar question. The plant height, canopy, transpiration, photosynthesis. And a big problem today of the modern machine is how to translate the massive amount of data we get to practical decisions. So before I will address these questions, I would like to address the challenges that we have when phenotyping plant environment introductions, also known as G by E, genotype by environment interaction. This is the biggest challenge we have, to phenotype the plant response to the environment. And I would like to explain why. So when we look at the plant, even under well-irrigated condition, the plant always sends many dynamic differential conditions. For example, the atmosphere. Sunlight, temperature, humidity are continuously changing and impacting the shoot. The shoot responds to the atmosphere, but at the same time, the water in the soil are responding to the shoot, which impact the root, which impact the water. And of course, all is connected to all, which means we have a very, very dynamic environment. So the challenge number one that we have is a very dynamic environment. Moreover, when you look, I took this slide from Plant Physiology, the 5th edition. I think it's also in the last one. So when you look on a plant stress response, so this is on the x-axis, this is the plant stress, you can see that we have hierarchy of responses. So first the plant biomass stopped growing, but at the same time, the plant stomatal conductance is still pretty good. And the same is for photosynthesis, which means when you phenotype the plant stress response, you always have to remember the second challenge, which is you want to have a direct measurement of multiple functional traits. Try to take both biomass trait and physiological trait because they respond at different hierarchy. So when we look at about this second challenge, we know that we want to take shoot traits like transpiration, biomass gain. We want to take root trait like water fluxes. We want to measure the atmosphere, the water in the soil, and so on. Basically, what we want to characterize in the G times E is the plant-water balance regulation. But balance, as it sounds, it's very hard to phenotype, okay? Try to imagine you need to phenotype this bicycle triangle, right? It's not going to be easy, and it's have to be continuously. I would like to explain the importance of this continuously measurement. This graph here shows you the stomatal conductance, okay? This is under non-stress condition, under well-irrigated condition. This is how a stomata behaves. In this paper, Broderick and Holbrook actually measured the stomata every few minutes. And you can see that between 7 and 8, the stomata increase conductance by 100%. Then when VPD goes up, this is the atmosphere, the stomata reduce transpiration, and several lines at 11 a.m. actually close stomata almost completely, which means that if you come and randomly measure your plant, some of them at 8, some of them at 9, some of them at 11, you're going to get to a very wrong conclusion unless you really do it simultaneously, okay? And if you do it on another day, when the VPD is much lower, it looks like a very different plant. So a continuous measurement and simultaneous measurement are very, very important. You want to measure all your plant simultaneously and continuously. So I would say this is challenge number 3 and challenge number 4. When you compare plants, and especially when you compare many plants, when you do high throughput phenotyping, as more frequently you do your measurement and continuously, which means for every few minutes in a day and for many days continuously, this will be better. So as more is better. Now let's talk a bit about drought. When we measure the drought response, we must remember that drought is a very, very dynamic trait. Let's look at this graph. Here we see many plants measure under similar condition. This is the plant atmospheric condition, light and VPD. This is the growth rate of the plant. Each color here is a different plant, and you see the water consumption. Each graph here shows the water consumption. I will explain later how we get it. Now, when the drought starts, you see a very big variation in the response of the different plant. So after 7 days, some plants actually under severe stress, while others are not stressed at all. So we can just say we close the tap here, but look what a huge variation we have in the treatment. So challenge number 5 will be when you do drought stress or any stress, abiotic stress, you must remember that you want to have a multiple and comparative treatment regulation. So we have to find a way to control this drought. So this plant and this plant will see not just similar ambient condition, but also similar stress condition in the soil. Different plants transpire differently. They have different sizes. So not wonder that they actually produce this big variety. So this is the next challenge, to find a way to control this droughted in a way that you will know what is the drought point. What do I mean by this? Drought point is a physiological parameter. Here, when I expose my plant to drought, I don't exactly know at which part on the graph the plant actually start to suffer. Maybe here, maybe here. Each plant actually start to respond to the drought at a different point. This point we call the drought point or the theta crit. Theta is the soil water content, which the water become limiting factor for transpiration. So if you look on this graph, on the y-axis we have plant transpiration, and on the x-axis we have soil water content. And you see, for example, that sunflower, okay, close stomata, this is stomata closure, this is the theta crit, at 40% of soil water content, while tomato, which is much more isohydric, will close stomata at about 60% soil water content. So determining the stress by the soil water content will be a mistake here, because if I choose 50, for example, tomato will be under drought, but sunflower won't be. Let me show you another example. Let's say I'm grafting a shoot, let's say tomato shoot, on different rootstocks. So if I measure now transpiration, this is again transpiration. So then under well-irrigated condition here, I will see similar transpiration rate. Now let's say that here I will reduce the water in the soil. So up to a certain point, both plants will transpire the same. But P1, the small root plant, will be limited faster. Although both pots have the same water level here, I will see P1 respond faster, closing stomata faster. Why? Because its root cannot reach all the water in the pot. So these two plants has the same amount of water, but P1 reach less. So you can say that his limiting factor is its root size. While P2, although have the same water amount in the total pot, will continue transpiring for a longer time. So we can use this drought point as a system to select root activity. Now, this is important to remember because usually we don't take a look on these things, and these things matters. So point number 6 or challenge number 6 is standardization. We have to know what is the drought level in the soil. We have to know what is the drought point of each plant. Only then we can really control a truly experiment which actually tests drought. So standardization, I think it's a challenge, not just to me, but to all the community of phenotyping. I think we have to find a way for standardization, our method, in particular drought, which as I show is highly dynamic. So what do we do in our system? When we do drought, in order to standardize the treatment, we actually continuously measure stomatal conductance. I will soon explain why, but we measure many parameters. Stomatal conductance is one of the most important, I think. We measure it under optimal condition. We expose the plant to drought and we look for the theta crit, for the breaking point, always in compared to control plants. So we can always compare the drought response to control. The delta between the two will be the yield penalty prediction. So we measure the drought response and we also measure the recovery, as I call it before, the resilience. And from this point, we actually compare to other plants, for example, this lower transpiring plant under optimal condition, but also this plant has lower penalty. And only if you compare all of them under all the conditions, you can really see these differences. Of course, some plants will suffer, so they will be susceptible. And we are looking for this line, which is the idiotype. I cannot say I find one, but at least I keep looking for this one. And each one of these plants going to have a different theta crit. So probably which lead us to also different yield. So all in all, when we do standardization, we do in vivo simulation. I will show you soon how, to predict the yield, and we try to do it in short period as possible. Usually it takes us 3 to 4 weeks to really do this prediction right. The seventh challenge, challenge number 7, it's the pot effect. This is a big challenge. Most of us, when we do the pre-breeding experiment, we do it in pots. We want to do it in the greenhouse before we go to the field. Nevertheless, working with pot leading to many problems like small pot size is fast drought buildup, salinity buildup. We have water loss by evaporation and so on and so forth. This was published, so I'm not going to get into details, but we must take care for the pots. We must remember pot may enter noise and we must take care of that. So the challenges I mentioned was the plant dynamic response, the fact we have to do direct measurement, continuous measurement, and simultaneous measurement. We want to have automation to control the multiple treatment, in particular drought, so we know exactly what is the treatment the plant gets while it's under the stress. So to prevent the fact that bigger plants will transpire more and get drought faster. We want to have standardization, and we want to minimize the pot effect. We also want to have these 3, I didn't talk about 8, 9, and 10, so I will mention now. We want to have multiple repetition, okay? Statistics is very important. So for each plant, we want to have, in our lab, we do at least 5 or 6 even repetitions, biological repetition for each treatment. We want to have a truly randomized experimental structure, okay? We don't want to take the environmental effects, so randomization is very important. And we also want to have a very simple tools so we could be, so we will be able to analyze the data as quick as possible and as accurate as possible. And I will talk about challenge number 10 soon. So as Philip asked me just before my talk, how did I get to this phenotyping business? I mean, so when we started a few years ago, I mean, it was more than 15 years ago, when we faced these challenges, we wanted to buy a system that will help us do the phenotyping, but also to cope all of these challenges. And unfortunately, the system that was in the market could not do all of this. I mean, there are very nice phenotyping systems that give you very good result about the plant morphology. There are very good systems that give you a lot of biochemistry, molecular biology. But as for the physiological part, well, we didn't find anything. So we built one for ourselves. We built a functional system, which later on created the plant detect system. So this system actually is a physiological phenotyping system. It was established in the Hebrew University, as I told you many years ago. We started by basic research. So we first recruited basic research money. Then when we saw it's really working, so we could, the Hebrew University had patent and we could get some seed investment. And then the company came, and in 2017, the first product was actually in the market. And from then we can see a few universities and companies all over the world actually using this system. So what is this functional phenotyping system? So basically, functional or physiological phenotyping give us the plant performance analysis of many physiological traits and help us to identify the ideotype plant or the ideotype behavior very soon after the experiment start, and in particular on the challenges I showed you before. This is actually a picture from our greenhouse, from the ICOR system in Israel. So we can see that we can measure many, many plants, actually hundreds of plants simultaneously. We can create many, each plant has many, many sensors. This is why we call it also telemetric system. So the sensors are combined to each plant and continuously monitoring each plant separately. The fact that we can control irrigation for each plant separately provides us the tools to build a truly randomized experimental structure. So we can really, really work with a randomized system. So each plant gets the treatment I wanted to give, and I can make as much repetitions I want. So we measure the soil, the plant, and the atmosphere. So each plant is like a resistor. Okay, so each plant is actually, we measure the flux of water through the plant. All the data goes to the cloud. We don't need any big services. The analysis is done in this controller that you see here on the spot, go to the cloud and bring me the data. So we get the soil, plant, and atmosphere data continuously and simultaneously on all the plants. So how does it work? Each plant has its own plastics. This is part of the patent of the company, which enable us to solve many, many of the pot effect that I showed you before. Each plant has its own controller. This controller measures the soil sensors. You can put up to 3 sensors per pot. The atmospheric sensors, you can put VPD, light, humidity, PAR, and so on, sensor up to each pot. And the irrigation. So this controller actually controls the irrigation from one end and measure the soil plant atmosphere continuum. So the control itself has 2 valves. Why do we need 2? Because many times we want to do cocktails. So this tab can bring one solution and this tab can bring other solution, and each plant getting its own cocktail of solution, either tab A or B or both. So if you look, this is the controller, this is the real controller software. So you can see that we can control the either tab A or B by time. This is simple, by weight. So we can use the fact that each plant is on a lysimeter. I will explain soon about this lysimeter, weighing lysimeter. We can use the previous day transpiration to irrigate. So we can tell the system, okay, please irrigate the plant and bring back the water it transpired yesterday, or use this control plant to transpire, let's say 50% from the control plant, or use some kind of the hydration rate. This is a very important trait when you need to do drought. So this trait will keep it in a rate of drought you want it to be. And you can also use sensor to control the system. For example, you can use commercial sensors, and you can say if you want to keep the system above or below a certain point. So this system helps us to solve all the pot effects. As I told you, this was published, so I'm not going to get into details. And also to solve many of the simple lysimeter system. So as I told you, each plant is actually standing on a lysimeter, but not, so maybe a few words about lysimetry. Lysimetry, or weighing cells, is a very old technology. It's more than 50 years in the market. The problem with the old lysimeters and data loggers, that they have a lot of errors, built-in errors that we have to solve when we build the system, we have to solve. One of the biggest challenge we have to face is the fact that a lysimeter is like a weight. When you stand on the weight, like your bath weighing system, you just get the weight of the whole of you, right? This weight cannot separate your muscles from your water, from your fat. But this is exactly what we want to do from our plant. We want the system to separate the shoot from the root, from the soil, from the water, and oddly Zymeter couldn't do it. But actually, the Plantarray, using this algorithm, controller, and plastics, enable to really do this. Today, we can separate the shoot weight, the shoot transpiration, the stomatal conductance from the root flux, from the soil type, from the water in the soil, and so on. So all in all, the system can control each plant separately. So you can really control a randomized, truly randomized experiment. It's measure the plants, analyze it, and help you to select the relevant plants. All the data go to the cloud and you can really see it in real time. What are the main measurements the system takes? So we take the whole plant biomass gain, transpiration and transpiration rate. This can be every minute. We have the water use efficiency of your plants, stomatal conductance, root fluxes, drought point, and of course, all the other environmental parameters. If you look on this small cartoon, so you can see the plants growing, each one grows under this other its own controller. And at the same time, we can see in heat map and histograms, the plant response. So we can make the treatment and also at the same time, follow the plant's real-time response to the drought, recovery, and so on. All of the algorithm and data that I told you was published, so I'm not going to get into details how we do all this measurement, but you can easily go to this technical advanced plant journal paper where we explain all of these methods. We call it high-throughput physiological phenotyping screening system for characterization, the plant-environment interactions. So after, I hope, explaining you all the challenges that one have to cope, we still have one question, how to translate the data to practice. So actually we use the SPACT analytical tools also from Plantarray, which help us getting a real-time statistics. So we can really see the treatments. So the system help us control the treatments, but we can also get statistically, for example, a ANOVA test or t-test, and we can really see if it's significant or not. We can set the alpha value and get data in real time. This is a very important parameter because it helps us to see and select the data in really fast time so we can actually push more experiment per period of growth. Let's see some data. So this is 1 day. This is the light regime in the greenhouse. This is the VPD, vapor pressure deficit, or the red line. This is what the atmospheric condition the plant sees. This is the lysimeter weight. So you can see this is one plant per 1 day. You can see 600 water loss. So the daily transpiration is 600 ml. We can translate it to E. E is the mole of water per mass plants. We normalize to mass. It's very important. I will explain soon why it's important to normalize to the plant biomass. And if we look on the stomatal conductance, which is basically the transpiration divided by the VPD, we can see the golden hour. This is a very crucial time of the day where the plant is much more efficient in its water use efficiency. So here the stomatal is much more open than at noon, while the plant transpires much less. The light is very efficient. So this point, as I will show you soon in some graphs, is very, very important to select plants' response to stress. Of course, so we can do what we call drought stress. So we can give, now you see averages. So this line is now average of few plants together. So we can stress the plant to either 80, 40, or 30% of soil water content. And you can see this peak, this golden hour. You can see it decrease during the drought. But even at the lowest drought, we can still see this peak rising. And actually, and this was also published, so you can see the details in the paper. But actually, these peaks are very, very important to select plants of better production in drought. In fact, higher peaks here are very good predictor to better yields under drought. We had many papers of the system, and I'm not going to get into details now, so you can just go to the literature and just find it. And I want to show you some result we have trying to look for the many traits I show you and to find what are the beneficial traits which actually help us to predict the data, okay? This experiment done together with Professor Dani Zamir, a very good geneticist researcher in my lab that also do field experiment with tomato. So Danny gave us data from a field from few lines, which has very different yield results in the field, both under good condition and drought. And we monitor their physiological parameters of the same plants. We saw very nice physiological characteristic that identify this plant. And then in order to get more repetition, we just do grafting of these plants, reciprocal grafting on these plants on themselves. Again, we get very nice differences in yield and physiological parameters. By the way, this field experiment was repeated for at least 2 years in the field. And then we did correlation with many, many physiological parameters and field parameters. We screen for the correlation, but most importantly, also for the significance probability. So we took only the high correlation and the significant probability characteristic. We go over all the parameters, and all in all, we select few of the important parameters. I will just show you few examples. For example, transpiration was very nicely correlated with yield in the field. So this is yield in the field, and this is the system parameters. You see both under drought and well irrigated, we get nice correlations. We also, fruit weight also give us nice correlation. Green fruit doesn't work so much, okay? Plant weight in the field have also very nice correlation. All in all, I will not go through all, I just want to give you a taste. So as I show you, when you work on plant fresh weight, the data from the system, especially the cumulative transpiration, which means the sum of all the water plant transpire during the season on the system, give us very good correlation with very nice probability. So very low p-value. But even more importantly, yield prediction, actual fruit in the field, was nicely predicted by stomatal conductance at the golden hour, between 7 and 10, again with a very nice p-value. So a significant correlation here at this point, at this GS. So all in all, this data show us that a particular time of the day for particular plants has a very good potential to be a good predictor. This is only 1 day, okay? This is only 1 day, so it's enough to take few days under well-irrigated condition, under drought condition, and at the recovery condition. And this is exactly what we did. We took these parameters under the well-irrigated, under the drought, always compared to control. As I told you, it's very important always to compare your plant response to a control non-stress plant. And at the recovery, for example, you can see these two plants that actually have the same transpiration, but very different yield. And the reason here is the recovery rate. Actually, this plant could not recover from the stress, while this recovered very, very nicely. So to conclude my talk, I talked today about this comparative functional characterization. So it's important to select your line. So for example, we can work with high productive line, medium productive line, or low productive line. It's okay. As long as you have the control, it doesn't matter. You're always going to compare your plant to its control. Like line A will always be compared to line A, and B to B, and C to C. Of course, you can also compare all of them one to each other. Now, when a stress starts, and I consider stress looking at the water content, let's say drought stress, the stress point will always be the point, not where the stress really starts, like here it starts here at week 5, but the stress will be defined by the theta crit. So although these plants transpire less, here I measure transpiration, for example, or GS. So the theta crit for this plant is faster. It doesn't matter this plant transpire less. This plant, for some reason, has a lower theta crit or a lower soil water content, which limit transpiration, while this plant has higher one. Either way, this response curve is only from the theta crit on, and then we also need to measure the recovery rate. So the recovery always will be compared, as I told you, to the control. So always our definition of control, resilience, tolerance will be compared based on the plant yield and recovery compared to its own control plant. Now, some plant transpire so less, like this line C, that they're never exposed to drought. So many people mistakenly take this plant as drought resistant. They are not resistant, okay? They pretty much survival. So if the drought was much longer, maybe then we'll see some decrease. So remember, soil drought is only when the soil limits the transpiration, but if the plant is very small, it's never been limited, okay? So all in all, you can do this controlling to do a short drought period or longer drought period, or in fact, terminal drought period, doesn't matter. It's always have to be considering the same standard. So if you repeat the experiment, you have to take in account the theta rate, the points I show you, and also the drought period. So to conclude my talk, I talk about the practical tools for G times E characterization. I talk about the important need to do in vivo simulation to put really plants in the size that usually represent crops, like we normally use 4 to 8 weeks, which as I show you gave a very nice prediction. Take the data simultaneously and continuously. Look for beneficial traits, which are, use some statistical tools to actually find this trait, like I show you this morning GS normalized to plant biomass. And all in all, the $1 million question will be, how do I know which horse is going to win the race, right? We want to put the money on the winning horse. So what I'm telling you here is that if you want to know the winning horse, build a simulation and really run horse in it. So if you want to find $1 million seeds, and this is the problem we usually have, we have millions of seeds, do a functional test, which is pretty short, usually take only a few weeks. Do it in vivo with real plants, get conclusion and throw away the ones that are going to fail in the field, and only then go to the field, see the data, and then improve your next selection, and so on and so forth. Okay, this is it. I just want to thank Sanbon Gossa. This is my PhD student that did most of the research result I show with the prediction. Professor Dani Zamir and his PhD student, Amit Koch, which actually worked with us on this prediction experiment. They actually gave us the data from the field and did all the field experiment. And Itamar Shenhar, my master student, that also contributed a lot to the data I showed here. And to my colleague, Professor Roni Volach from the Soil and Water Department in the Hebrew University, which actually many years ago started with me the Plantarray system and established all the algorithm I show you today. And thank you for listening to me.
Thank you, Menachem. Wonderful talk. I mean, I've seen it now, I think, more than twice, and I must say it's a very, very nice depiction of almost of a breeding pipeline using transpiration metrics together, of course, with this optimally suited system that you have created. Very nice, very relevant, not only in Israel, not only in the greenhouse. And there I would also start with my first question. I mean, most of the studies you have shown and most of this research you have done in greenhouses. Of course, your system is optimally suited for greenhouses. However, at some point during the process, you have to test whether the selected lines are also performing in the field. So how transferable have the results obtained in the greenhouse proven in the field?
Yeah, so this is really the $1 million questions. So for tomato, as I showed you here, we get very, very nice correlation to the field. So we get up to, I will go to the table here. So we get up to sometimes 90% accuracy with 0.04 p-value. Of course, sometimes you get also 0.3 or 0.4. it was important to me to show you all the data. Today, we start to investigate with a lot of AI tools in order to find this really, because I want to get to this 90% very fast. So now we actually conducting a huge experiment in the field. We know that to predict the biomass, so if you work for let's say, greenies, like, I don't know, like cabbage or lettuce, we can get up to 90% accuracy with 0.01 accuracy. So if you, the main thing I would say, solve the potting effect. If you solve the potting effect, as I explained, especially the size of the pot, because usually when you do drought in pot, you get accelerated drought. Let's say if the plant will reach the field, the tetacrit, after 5 days, in a pot it could be 2 days. So this feedback irrigation we use really help a lot to reduce this pot effect. We also have some other techniques I don't have time to explain, but we actually published this. So solving the pot effect, I would say this is the #1 criteria to overcome a lot of noises that usually give you malpredictions. But of course, also other conditions, you know, each year look different, and sometimes one year you get good prediction, the second year you get bad prediction. This is why the results I show you here are at least from 2 years. Now we have another study for 4 years in the field. Of course, as more years you have in the field, the better your result going to be predicted. But I would say this, plant physiology are very robust measurement. If the plant has high vigor and high transpiration in your pot, usually at young age, you will see it in the field as well. The question, if you can really build the experiment that mimics this correctly, I would say this is the biggest challenge. But if you do it correctly, your prediction will be pretty good.
So in that sense, is there any ambition to adapt the Plantarray system, for example, to use bigger pots, mesocosms, or maybe even a field version? Is there currently something in the pipeline in regards of innovation?
So yes, I don't know if I have a picture here. We do have bigger pots. We have 40 liters. Oh, you see these pots in the picture where you see the sorghum grows? These are 22 liters. The same thing, only bigger pots. We can put trees. We actually publish, I can show you here papers about pine trees in Israel. So this tree physiology paper, we actually grow pine trees for about a year in this system, and we get very nice ecological responses. We want to go to the field. This is also thing. So now we adopt, I also have, I think, a figure here. Where did I put it? Oh, it's in the end. So now we adopt new sensor in order to have sensor that are much more accurate to the field. I will show you in a second if I find it. Oh, so now we use new NDVI, PRI, and other sensor in order to have a non-lysimetric, because to take the lysimeter to the field is a big challenge. But there are other parameters that may work very nice also in the field. So our new version of the Diagnostics system give us a lot of in parallel measurement. So you can actually measure these parameters on the pot, and it will be much easier to implement it in the field in the future.
Yes, that was also one follow-up question. How do you calculate the drought point in the field?
Yeah, in the field, it's going to be a challenge. But again, I would always say, go to the physiological parameters. So if you can measure, for example, if you have a good way to measure stomatal conductance is one of the more sensitive drought point traits. Stomata will respond to drought quickly, very, very quickly. So if we find a way to measure accurately stomatal conductance, then I think it's a good point to test the tetacrit. I must say we didn't do it. I cannot say that I have the right way to do it. It's still under process. But you know, today we talk about the corona and people ask what is a bad patient, patient with a strong condition or bad condition. So at the beginning, the doctors say, okay, if it's walk, it's probably good condition. And today they take the oxygen level in the blood. So only oxygen level in the blood will tell you what is the severity of your disease, right? Even if you run, but if the oxygen level are slow, you need to go to bed. So this is the main idea of the physiology. Measure the physiology. The plant may look good, but it may be very suffering.
Yeah, nice analogy. So one question's popping up from Olumidea, and he or she is asking, using the biomass as a correlation factor in the measurement, what about plants that have a nice vegetative growth and less fruiting? How do you correct for that at experimental level in this kind of experiments?
Very, very good questions. I'm glad. I would like to show you what we did. So we divide the experiment to two phases. It's actually two breeding goals. So one phase, it's indeed, let me find a slide, please, just a minute. So, yeah. So as I explained here, we call it resilience, which is the vegetative phase. This is why I suggested to call it resilience. So I suggest that if your plant has a very nice biomass, it has a nice recovery rate, so it has a good resilience. But you also have to see what is the yield. We do counting of flowers and fruits and see the ratio between the two. So if the fruit load is low, you have a low tolerance. So you could have high resilience with a low tolerance. And what you really want to have is a high resilience with a high tolerance. And in fact, these are different characteristics. It's different genes, it's different breeding goals. In fact, you can find a plant with a good resilience traits and try to cross it with a good tolerance rate. So it's really two aims. And I'm glad that I've been asked, because it's important to note the vegetative stage is totally different than the fluorescent stage. And I think I gave the example before, but I will repeat it. Maize, it's an excellent, excellent example. Maize have a single shot chance of flowering. You know, so maize, pretty robust plant, is a C4 plant. Now, if maize is going to be exposed to drought during flowering time, the chances is going to lose the flower. And then doesn't matter how fast the plant will recover. It's not going to have a second flowering part. So the plant going to be nice and green, high resilience, but very low tolerance. And if you look on CIMIT papers for the last 30 years, you can see that most of the successes in the drought, maize drought breeding was in the female flowers and in the ears. So plants with shorter time from flowering the male to the female actually has the better stress tolerant in the field. So I hope I make myself clear, but this is clearly two traits to look at, two different traits.
So Menachem, you have been mainly working as on a model crop, on tomato. So have you had similar results in other crops as well, or are the mechanisms, do they differ quite a lot between crops? I mean, I have heard about similar observations for sorghum, for example, also using plant transpiration. But what are your experiences?
So yeah, you're right, Philip. We mainly work with tomato, and in tomato we find this highly correlative. Tomato have this ability that even if it's hurt under flowering, many times it still have another round of flowering, but I cannot say too much about cereals. We work a bit with barley, but this was wild type barley. We had a big project of looking for a resilient trait from Israel wild types. You know, Israel is the origin of barley and wheat. So we look for many cultivars from Israel and we look for resilient traits. But no, we don't have too much data about other crop for now.
Mm-hmm. And in terms of applications for the Plantarray system. I was wondering personally if there has been any particular interest from people working in controlled environment agriculture up to maybe even vertical farms that where I can think of that a system like yours is quite useful. Have you had any inquiries from that side?
Oh yes, many. Now I say number one is the cannabis industry, which many times it's, so we already working with them. Cannabis is an interesting plant because there are not too many papers, you know, for many years it was not legal and now it's become legal in many countries, but people don't know basic stuff about how to irrigate, how to fertigate. Many questions. The response to VPD, something that we know in tomato for like 50 years, you don't know anything. So now we have actually more than one research going on this plant. Vertical farming is also in a big rise now because people want to use better the places on, you know, on the rooftop and the urban city. So we have a lot of interest from this field as well. It's looking like, yes, it's looked like a very fast-growing area right now.
Yes, it is. So we'll see what that comes in the future. And from our side, IPPN's emphasis and also EPPN 2020 side, we wish you the best for the future, for both your research and your company. Once more, you will find, so this is going now to the audience, you will find all the relevant links, both to Plantarray and to the iCore system in the chat. Be sure to note this down, but also feel free to contact IPPN on any questions or in queries in the direction of Menachem, and I will gladly channel this through. Menachem, do you want to say anything towards the end?
I hope they find vaccine to the corona soon.
Okay, then I would quickly tell a few things at the end, mainly going in the direction of the next Phenomics webinar, which will be in about 2 weeks. Let me quickly show a slide. So next week already, there will be the release of the 4th IPPN newsletter. So far, be sure to check on all recent news around the global plant phenotyping community and IPPN as an organization. And part of this newsletter will be the Global Plant Phenotyping Survey for 2020-21. Usually, the Global Plant Phenotyping Network, together with Emphasis, the European partner, is releasing these kinds of surveys in a biannual manner. And this survey has the purpose of giving us information on the current status of plant phenotyping globally, based on where you are, what profession you have, and in what field you are. This information is then in turn being used to guide our actions, to see where actions are necessary, where collaboration can be fostered, where industry as well as academia can be can be helped and facilitated in the plant phenotyping space. And then, so be sure to check this. I will also post the link in the comment section, but feel free to just visit our IPPN page and check the newsletter subscription there. And then in 2 weeks, we will have our last Phenomics webinars for this year. Then we will have a short winter break, and then we'll start anew in 2021. Maybe Menachem will be part of our 2021 setup. who knows? And we will gladly be accommodating him once more. Thank you, Menachem, once again.
Thank you very much, Philip. It was a pleasure.
Thank you. And be sure to check the last Phenomics webinars, which will be a session on field phenotyping. Very good. Thank you very much and all the best. Have a nice weekend to come and see you next time. Bye-bye.
Bye-bye. Thank you.